Hello, everyone. So let us continue our discussion on infrared spectroscopy. So in the previous videos, we look into how this infrared radio, uh, uh, infrared spectroscopy is actually how does it look like? So if you see here, the infrared spectroscopy, it basically consists of uh, plot between percentage transmittance and the wave number. Okay, so in this you will see a lot of peaks coming in the spectrum, and these peaks correspond to the basically the absorption bands. That means various bonds absorb different frequencies. Depending upon which frequency has been absorbed, that will be coming as a peak in the spectrum. Okay, and an important thing here to note is that the different peaks that you see here have different level of intensity. Okay, some of the peaks like you see here, these are kind of weak peaks. Okay, however, when you come to peaks here, uh, this particular peak, if you look into, this is kind of a broader peak, okay, having a higher intensity, okay. And these peaks looks like some medium level of intensity peaks you can see here. So in the IR spectrum, usually we see peaks with varying intensity. Some of them are pretty stronger intense peaks. Some of them are kind of a mid-range of intensity. Some of them are like having a very low range of intensity. Now, what is the basis, like on which factor is this intensity dependent upon is what we are going to look in the today's video. So basically you have now understood the various how it looks, and the previous video I also gave you a table that talks about the different types of bonds and their corresponding frequencies at which they appear in the IR spectrum. And as you can see here, each of these functional groups and their corresponding frequency, they have different level of intensity that they shows. Like, for example, for a particular consideration, let us look into CO. CO has a kind of range of frequency around 1780 to 1650. Now, if you look at the C double bond C, okay, this kind of overlap with this particular region. Okay, it's 1680 to 1600, and it's basically 1780 to 1650. So if the peaks of the CC bond, C double bond C bond, and C double bond O comes, often there is a chance that they overlap with each other, or they might look similar to, they might appear close to each other. Then how to distinguish whether that peak is corresponding to C double bond O or it is corresponding to C double bond C. See, if there is only C double bond O present in your molecule, then you don't have to worry about it. But if suppose you have an alpha beta unsaturated molecule where you have a C double bond O as well as C double bond C. So in that case, to identify which peak correspond to a C double bond C and which correspond to a C double bond O, here this factor of intensity come into the picture. So in this table, if you look into the C double bond O, the peaks are pretty stronger. However, when it comes to C double bond C, the peaks are of a medium intensity. Okay, so that's why in understanding of the factors on which this intensity depends on is very, very important. So that is what we are going to now look into, like on what factors are this intensity dependent upon. So basically intensity is dependent upon three factors. One of them is the change in dipole. Okay, another is the number of bonds responsible for the absorption. Third is the concentration of the sample. Okay, now what do they mean? So basically, as I already gave you what intensity means, so it's basically the area under the curve. So if you have a very intense peak, that means the area under that peak would be pretty high. But if you have a less intense peak, the area under it would be pretty low. Okay, so The area under the curve of the peak gives you an indication of the intensity. Now, the change in dipole moment. Okay, so if I ask you, let us continue with with the example that we took into consideration. If we compare CO and C double bond C, if I ask you in general, being organic chemistry students, like which of these molecules is having a higher dipole moment, then you will quickly say that it is C double bond O. Okay, so when you have a higher dipole, okay, like in the case of C double bond O, then it will give a larger intense peak because the change in dipole moment is pretty significant. Because as you have studied in molecular spectroscopy in your physical chemistry class, you know an important fact that for a molecule to be IR active, there should be a presence of double bond. If you take homoatomic molecules like H2, Cl2, all these molecules do not have any dipole moment, so they are IR inactive. So for any molecule or any bond to show any kind of infrared activity, it is very essential that they should possess a dipole moment. So C double bond O and C double bond C, if you look into consideration, C double bond O does have a dipole moment. C double bond C also has a dipole moment depending upon the groups that are attached to the C double bond C bond. Okay, so in this case, since we are considering that both of them has some degree of double bond dipole moment, so they will be IR active. In that case, if we compare the dipole moment, obviously C double bond O will have a higher dipole moment. So change the vibrational, when the vibration happens in that C double bond O bond, the change in dipole moment is significant. Okay, depending upon the formula, the distance and the charge, when we take into consideration in dipole moment, when the distance changes or any variation happens, the dipole moment change is pretty significant. So that is why, as a result, the corresponding peak that is associated with this particular bond would be pretty much intense. Okay. In the same way, if we compare similar kind of bonds like OH, NH, CH, it's very easy to now say that out of them, OH would be very intense, then followed by the NH bond. CH would be the least intense bond. Okay, so we can easily find out from an IR spectrum, though they are falling in the range of 3000. Okay, you can easily distinguish which peak is CH, which peak is NH, which peak is OH, especially in compounds where you have all these three present. Okay, in such case, you can easily identify, depending upon the intensity of the peaks coming in the 3000 region, which peak correspond to OH and NH and CH. Usually OH peaks are pretty easy to distinguish. As I just now showed you, they are pretty broader peak. It's not a sharp peak, it's usually a broader peak that comes into the picture. Okay, so that is one factor that decides upon the intensity of the peak. Another factor that is crucial that decides the intensity of the IR peak is the number of bonds that are responsible for absorption. So if you see here, if we compare the intensity of the CH peak in the octyl iodide, okay, and if you compare it with the methyl iodide IR spectrum, the CH bond that are present in octyl iodide, there are 17 CH bond present. You can easily draw an octyl iodide, okay, and in methyl iodide, there are only 3 CH bonds present. So if you compare the intensity of the IR spectrum of this, and especially look into the 3000 region where your CH bond of CH frequency, the CH vibrations happen, there you will find that the peaks corresponding to octyl iodide would be pretty intense because their number of CH vibrations that are coming is more. So they will add on, therefore the intensity appears to be pretty much bigger when it is octyl iodide in comparison to methyl iodide. So another factor on which the IR intensity depends is the number of bonds that are contributing to that particular peak. So if there are more number of bonds present, it would make the peak more intense. In a similar note, when we talk about concentration of the sample, if the sample is concentrated, obviously the bonds present would be larger in number. So that also can lead to a pretty more intense absorption. So often when we do some kind of experiments in the lab and we get samples in a very small amount, okay, and we try to give the IR of that molecule. Since we have a very small amount, we give a very small amount for detection, because once we give the sample, usually that's gone. So when we give it for detection, what we find is that the peaks are pretty less intense, because we have given them a very small portion of the sample. So often if you require a very good IR spectrum, you need to give some minimum good quantity of the compound. Only then you will get a pretty intense and clear peaks on the IR spectrum. That doesn't mean that you have to submit a lot of sample. It's usually in microgram, very small amount you need to give for IR detection. But still, depending upon the peaks that is coming, you can decide whether you need to give more sample or less sample, and so that you can get a more clear peak distribution. Often organic chemists, since they are only interested in the functional groups, they will just give small amount of compounds, because they are not worried about the fingerprint region. They are worried just about the functional group where the functional peaks comes. So they can easily identify, or when the functional group peaks that come in the functional group regions are pretty, even though they have a medium intensity, you can easily find them out. So for us organic chemists, slight less sample and the peaks being little less intense doesn't matter much in the usual cases. Okay, so that is the importance of intensity in an IR spectrum. So IR spectrum, an important factor that helps us in analyzing an IR spectrum, is the intensity of absorption. So depending upon the intensity, you can distinguish peaks that are coming at a very close range. Okay, so that is one important aspect in IR spectroscopy. So I hope you understood this particular thing. So in the next video, we will be talking about what decides the position of the peak that is coming. That means like if CO is coming at 1700, why it is coming at 1700? If OH is coming at a pretty higher frequency, why it is coming at a very higher frequency? So what decides the position of the peak in the IR band is what we are going to see in the next video. So I hope you understood this. Please go through more organic textbooks and you will understand better. Thank you very much for your attention.